Hi everybody, so you know alright the title. I'm talking about aquaculture. Aquaculture is a millennial-old activity that has evolved slowly, often by building on traditional knowledge. However, scientific progress in the 21st century brought an exponential growth. The production of aquatic animals in 2020 was more than 60% higher than the average in the 90s. Outpacing world population growth, and this is largely due to the increasing aquaculture production. In 2020, fisheries and aquaculture production reached an all-time records of 240 million tons. And this stunning expansion presents several challenges that must be addressed to achieve the final aim of sustainable farming. And sustainable aquaculture relies on multiple factors, including farmed fish, water quality, and feed formulation. Why I mention feed formulation? Because replacing fish meal with alternative protein sources is a key objective to improve sustainability in aquaculture and promote fish health. So instead of using fish to farm fish, it's better to decide to switch the protein sources using other protein sources. Insects appear to be a valid alternative to fish meal because they are rich in nutrients, have a lower environmental impact than the other plant-based protein sources, such as soy, for example, and constitute part of the natural diet fish, and this is also very important. One of the most used species of insects in aquafeeds is the yellow mealworm, the Tenebro molitor. You can see the larvae on the upper panel and the adult in the lower. A lot of research efforts have been made to evaluate a novel feed formulation, considering a lot of points of view, such as economic sustainability, environmental sustainability, looking, for example, at the carbon footprint or the water footprint. But considering the microbiome-related studies, they all focus especially on the effects on the fish gut microbiome. This is important, but this is not sufficient, because for a deeper and extensive comprehension of the aquaculture ecosystem dynamics, you should look at all the interactions that links the host-associated and environmental microbial communities. Our story is about a hen throwing feed into a flow-through tank in which toads are reared. And so we wonder what happens then. Imagine that you can see the characters of this story thanks to a molecular lens. Here we want to evaluate the influence of insect-based novel feed formulations on the environmental and on the fish microbiomes. What are the methods? We collect water microbiome samples, tank biofilm microbiome samples, feed microbiome samples, fish skin mucus microbiome samples, and fish intestinal mucus microbiome samples. We apply the classical way, sequencing the 16S03V4 region of the rRNA gene, using Illumina platform, the Illumina MySeq. We perform the bioinformatic analysis using Time2, and we use Silva as a reference database. The clinical trial was performed in a flow-through experimental fishery facility where a rainbow tooth Oncorhynchus mycis was farmed. Feed formulations consist of increasing percentages of inclusion of partially defected meal derived from Tenebro molitor larvae. So the diet A is the control diet with no insect inclusion, and the diet D is the diet with the highest percentage of insect inclusion. There are also intermediate diet, diet B and diet C. We collect water-related samples from all the feed formulations, but fish-related samples only from diet A, the control, and diet D, the one with the highest inclusion, to limit the sacrifice of fish. So let's go to the results. Our results, first of all, show that considering all the metrics, water samples, in particular inlet water, the water that is entering in the tank, had the highest alpha diversity, followed by tank biofilm samples. Instead, the lowest alpha diversity was measured in the gut mucus and in feed samples. When we look at the taxa bar plot, here is plotted at the family taxonomic rank. we can see that the three most abundant families are Mycoplasmataceae, Commamonadaceae, and Flavobacteriaceae. This is quite expected. And just looking at the colors, we cannot see any striking differences considering the feed formulation. But maybe we can appreciate some differences considering the sample source. So if it's water instead of mucus or feed, and so on. Then we applied a very simple analysis, a Venn diagram, and considering the sample sources. And here we can see the distribution of shared ASVs among the different sample sources. And what is quite nice is that there are only 8 ASVs shared by all the sample sources. And the highest number of unique samples, unique ASVs, are in water and tank biofilm sample sources. According to the NMDS plot drawn on Bricartis dissimilarity, we can see again that samples cluster by sample sources, and there is a clear cluster that is separated from the others, that is the one in upper right, that is made by feed samples. Again, if we perform a network analysis based on Bricartis, we can see that there are two subgraphs. The major subgraph, the one on the left, comprise the majority of the samples, and the minor subgraph, the one on the right, is again the one made by feed samples. And there is no differences related to the feed formulation administered in the trial. So it seems that different feeding formulations were not the drivers of the correlations of the microbiomes. Thus, to deepen the analysis and disentangle if there are any contribution of feeding formulation at a finer scale, we decide to perform a differential abundance analysis using negative binomial generalized linear models. And the results are that when we compare the two feeding formulations, the control, the feed formulation A, and the one with the highest inclusion of insect, the feed formulation D, considering all the sample sources, we found that the control, the feed formulation A, had the highest number of genera with a positive differential abundance compared to those related to the feed formulation D. So the control, the highest number of genera. And then we decided to compare only the water-related samples, so the tank biofilm samples against the water samples, again considering the feed formulation A and the feed formulation D. And when we perform this comparison, we observe that a higher number of genera are significantly more abundant in water than in tank biofilm, and also in feed formulation A than in feed formulation D. After that, we decide to compare the inlet water, the water that is entering in the tank, and the water that is inside the tank, to see if there is any contribution to the microbiome made by the fish that is reared there. And we see probably exactly this. So the tank water has a highest number of taxa that are more abundant than the inlet water. And so maybe there is a contribution of farmed fish in shaping the structure of microbial communities in terms of abundance of specific taxa. And this is quite important to try to figure out how to manage the aquaculture plant. So what are the take-home messages? First of all, the water ecosystem and the aquaculture are a built environment. So the aquaculture itself can be considered a built environment, an artificial environment with its own peculiar microbial communities. So also overall, water shows an astounding microbial diversity compared to host-associated and feed samples. And water sample, in particular the water entering the tanks, has the highest alpha diversity, followed by tank biofilm samples. This is not surprising because there are a lot of evidences that show that water ecosystems are highly biodiverse. Understanding the extent to which an animal's microbiome can be influenced by its surroundings, so the built environment, and then associated with a phenotype, such as fish health, will be important for experimental design where microbiome readout is a standard measure. The second take-home message is the aquaculture should be considered as a holobiome. So the inlet water represents the background microbial community entering the tanks. Feed is constantly administered to the fish in the tank and is carrying out its own microbial contribution, even with the low biodiversity. And water is the connection between the surface of the tank, the biofilm, and the fish, the skin and the gut. So all the analysis carried out, spanning from the Venn diagram to the alpha diversity, the beta diversity, and so on, converge in one of the hallmarks of aquatic ecosystem, the extreme biodiversity. And so the aquaculture plants are in this case. And the major driver of the microbial community structure is the sample source, not the feed administered. Even if we were able to show also that subtle but significant differences are exerted by the feed formulations. I want to conclude my presentation with some preliminary results because we wonder what about different insects for novel feed formulations. So if we change a Tenebrio molitor with another insect, what happens? We start considering water microbiome samples, and we tested this in a trial where Hermetia illusions, that is another insect, was used for the feed formulations. And here you can see from this PCOA based on weighted unifrac that there are no clear clusters considering Hermetia versus Tenebrio water samples. And also that there are a lot of families, 277, that are shared between Tenebrio molitor and Hermetia illusions clinical trials. So we should do other analysis, and maybe in the next talk I will tell you about these results, but for the beginning, these are really promising. So I want to thank you all, and in particular, thank the funders and supporters and the team that made this project possible. Thank you.